CADDIS is an online application, and it's designed to help users conduct causal assessments primarily in stream ecosystems. And Kate Schofield is going to be walking us through the CADDIS and showing us how to use it today. States such as Pennsylvania have used CADDIS to determine the causes of impacts to aquatic health, for example, in the Susquehanna River. If you have a question during or after the presentation, we ask that you please type it into the Q&A pod at any time. And once the presentation is over, we will answer as many questions as time allows. And we will be making the slides from today available on the EPA Research website. And Erin will type that address into the chat pod for everyone. So without further ado, our presenter today is Kate Schofield. Kate is with EPA ORD Center for Public Health and Environmental Assessment. Kate, thank you so much for doing this today, and I'll go ahead and turn it over to you.
Great, thanks, Lisa. Can folks hear me okay?
Yes, sounds good.
Okay, great. So today I'll be giving a presentation on CADDIS, but please understand that CADDIS really represents the combined effort of dozens of scientists and resource managers at EPA and in the regions and states over the past 2+ decades. So I really have a cast of, if not hundreds, definitely dozens and dozens of people behind me on the content of this presentation. This presentation is meant to provide a brief introduction to CADDIS, EPA's Causal Analysis, Diagnosis, Decision Information System. So it is a mouthful. The acronym is a reference to CADDIS flies, larval aquatic insects like the one shown in the top right corner here, which are generally considered to be indicators of good water quality. In this talk, I'll go through a quick overview of causal assessment, including how CADDIS, causal assessment, and stressor identification, 3 terms that are often mentioned together, are related. I'll then walk through a quick tour of the CADDIS website, and as I go, I'll try to highlight how CADDIS can be used in causal assessment as well as in other applications. So going back to those 3 related terms, causal assessment, stressor identification, and CADDIS. Causal assessment refers to a process to determine the likely cause or causes of an observed effect. Stressor identification is a specific method that was developed to determine the most likely cause of observed biological impairments in aquatic systems, primarily in streams. This process was initially documented in EPA's Stressor Identification Guidance document released in 2000. CADDIS, or the Causal Analysis, Diagnosis, Decision Information System, is a website that provides information, methodology, and tools to help users implement stressor identification and conduct causal assessments of biological impairment. I'll discuss the resources available on CADDIS a little later in the presentation, but it's first helpful to think a bit about causal assessment more conceptually. There are basically 3 different tiers of causal assessment. The first examines general causation, which is more theoretical in nature, and considers whether a specific cause, C, can result in a specific effect, E. For example, does or can smoking cause lung cancer in general? Does or can chemical Z cause lesions in fish? Contextual causation examines the conditions under which a specific cause can result in a specific effect. For example, does smoking cause lung cancer only when certain genetic factors are also present? Or does chemical Z only cause fish lesions when it's present above a certain concentration? Contextual causation starts to tease apart the key factors that affect whether cause-effect relationships are observed. Specific causation examines cause-effect relationships in very discrete situations. Did smoking cause lung cancer in Ronald Fisher? Did chemical Z cause fish lesions in my stream? These questions of specific causation are typically what we're interested in the CADDIS world, to address what's causing biological impairment in very specific individual streams or areas. But the methods and tools provided in CADDIS help us to apply information from general and contextual information from general and contextual causal assessments to these more specific cases that we're focused on, the CADDIS assessment. And as a quick side note, this is a picture of Ronald Fisher at the top right here having a smoke. Fisher was a scientist who studied causal theory and who famously opposed the idea that smoking causes cancer on the grounds that most people who smoked did not die of cancer. Ironically, he was not one of those fortunate smokers, and he did end up dying of lung cancer. So in CADDIS, our main focus is on specific causation, but why is this important? Biological monitoring and assessments are commonly used to identify if streams are impaired, and in many cases, the causes of observed biological impairments are unknown. So the table on the right here ranks the causes of impairment for the most recent data available on Clean Water Act 303D listed waters. Pathogens, sediment, and nutrients take the top 3 spots, but cause unknown groups come in at 11th, 12th, and 33rd place. And when you total all those up, all the miles in those 3 categories, you have roughly 90,000 stream miles that are impaired for unknown reasons. And the bottom line is that it's hard to fix a problem when you don't yet know what to fix. Stressor identification puts forth a formal method for getting at that question of what should we fix. Why is this necessary, though? Why can't we just kind of wing it and see what answer we get? A formal method is useful because we, people in general, can make a lot of mistakes about causality when we don't think about it correctly. We tend to form initial impressions quickly based on whatever information we have readily available. And this is the reason for the phrase jumping to conclusions. And this can result in a few things. We tend to overweight chance events, for example, by thinking that every time I wash my car, it rains. We can have biases that we bring to bear when we're thinking about things. So, for example, thinking that all pollution is caused by industry. We may be educationally predisposed. We know what we know, and so we tend to view things through that lens. For example, hydrologists tend to think hydrology, toxicologists tend to think toxics, and so on. And we tend to rely on intuition and past experience. If we've seen something similar before, we may be predisposed to see that same thing the next time. For example, if the last time I saw this impairment, it was caused by high nitrogen levels, it's probably nitrogen again. Okay, we also tend to gather information that supports our initial impression and ignore evidence that doesn't support our initial impression. This is referred to as hypothesis tenacity. Once we have a hypothesis we believe in, it's often hard to let go of it even in the face of conflicting evidence. And finally, we tend to confidently reach conclusions based on incomplete information, a cognitive bias described as wisiati. What you see is all there is. Given that we tend to make these mistakes about causality, a formal method helps minimize the effects of all these tendencies. To paraphrase this quote from Richard Feynman, we tend to be easy to fool. So by using a formal method for stressor identification, we're basically trying to reduce the chance that we fool ourselves. The approach taken, the causal assessment on CADDIS, which I'll get into in more detail shortly, has several good things about it. It provides that formal method, which allows for defensible and transparent evaluation of causes. It can identify causal relationships that may not be immediately apparent or that you haven't thought of yet. It helps to minimize bias and other lapses in logic. It helps to identify all available evidence that can be brought to bear on the question at hand. And finally, it increases confidence that whatever actions you decide to take based on the assessment can improve biological conditions. It increases confidence that you're going to be addressing the right problems. However, there are also some downsides to the CADDIS causal assessment approach. These assessments are not always or not even usually easy or straightforward. There can be lots of potential causes to consider, some of which you may not even be on your radar at the outset. The mechanisms driving biological impacts can be complex, and so causal assessment can get complicated quickly. The approach relies on data, so both data quantity and quality are important. Often data are limited, particularly when you're looking across many potential causes. Ultimately, causal assessment may not lead you to the smoking fish. You may not be able to identify that clear culprit or clear cause, or multiple stressors may remain as likely causes at the end. But this brings us back to the good part. Even when a single likely cause isn't identified through the causal assessment process, it often narrows the universe of possible causes, which helps point you to promising avenues for new data collection and analyses. The list shown here are candidate causes that were considered in a causal assessment of a fish kill in the Potomac and Shenandoah watersheds of Virginia and West Virginia a few years ago. We didn't get any answer in the initial assessment, but we were able to rule out 5 of the 9 original candidate causes considered, which helped to focus future data collection and analysis efforts on the remaining 4 possibilities. So we could stop thinking about some causes because we could show that those weren't in play, and we were able to focus on the ones we were still uncertain about. So this brings us to the end of the more theoretical introduction to causal assessment and stressor identification and into the nuts and bolts of CADDIS, which again is a website designed to help users conduct causal assessments of biological impairment. You can access CADDIS at epa.gov/CADDIS, and it'll bring you to the page shown on the right here. CADDIS is organized into 5 volumes: stressor identification, sources, stressors, and responses, examples and applications, data analysis, and causal databases. I'll dive into each one of these in a bit more in the following slides. The bulk of the content on the site was released in 2010, although there have been substantial changes, particularly in Volume 5 since then. The overall look and navigation of the site also changed substantially a couple of years ago when we converted the site to Drupal. So it does look a little different now than it did a few years ago. CADDIS was originally developed for causal assessment in streams, and so much of the information is geared towards stream systems. For example, the stressors CADDIS provides background information on in Volume 2 are common stressors in streams. However, the stressor identification framework itself and many of the methods and tools included in CADDIS can be used for assessments in different habitats, as well as for assessments other than causal assessments of biological impairment. For example, we just learned that the CADDIS stressor identification framework was used in a recent publication looking at hospital-acquired infections from sink drains. So those researchers took the CADDIS framework and adapted it to a completely different field. So these principles and tools can be applied more broadly, and I'll touch on this point a couple other times throughout the presentation. Volume 1 of CADDIS is stressor identification, which presents the strength of evidence framework outlined in EPA's 2000 stressor identification guidance. This volume contains a step-by-step guide to the stressor identification process, along with information on the theoretical background of causality and causal assessment. The step-by-step guide walks users through the 5 steps of the stressor identification process, circled in red here. The trigger for this process is detection of some biological impairment without an obvious cause. The first 2 steps, define the case and list candidate causes, are the problem formulation stages, where you identify and set the stage for the assessment. Once these steps are completed, you move into steps 3 and 4, where you dive into the available data and conduct the analyses that will ultimately get you to step 5, where you weigh all the available evidence to identify the probable cause. Volume 1 of CADDIS lays out the framework for these 5 steps, and then volumes 2 through 5 provide information, methods, and tools that can be used in these different steps of the process. I'll walk you through each of the steps in a little bit more detail. In step 1 of stressor identification, you define the case. This means clearly delineating what specific biological effects were observed, where and when did those effects occur, and where are those effects absent or different. And this last one is to identify sites, preferably nearby sites, that can be used as comparison sites in the assessment. Once the case is defined in step 1, You move on to step 2, list candidate causes. This is where you start to generate hypotheses about what's going on and begin to think about how you might evaluate those hypotheses. In step 2, you generate your initial list of potential causes, gather information on potential sources, stressors, and exposures in your system, develop a conceptual model that visually maps out hypothesized causal pathways that could lead to your biological effect. Then, based on all of this information, you develop the quote-unquote final list of candidate causes that you'll consider in the assessment. And final is in quotes here because it's often not really a truly final list. The list may get winnowed down again at some point, and often it's really a bit of a living list that's revised multiple times throughout the process. And this brings us to the meat of a causal assessment, the analysis phase. In steps 3 and 4 of the stressor identification process, you evaluate the data, both data from the case and data from elsewhere, and I'll come back to what each of those phrases means shortly. These steps are where the available data gets translated into evidence for or against your different candidate causes. Before we get into steps 3 and 4 specifically, it's worth talking a little bit about what we mean by evidence. What is evidence? Evidence is any available information that indicates whether a belief or proposition, or in our case, a hypothesis, is true or valid. Any information or data that help us form what is hopefully an accurate conclusion or judgment about something counts as evidence. And it's also often helpful to think about evidence in terms of if-then statements. If a specific cause X produced the specific effect Y, then we would expect to see result Z. For example, if we think low dissolved oxygen caused a fish kill, then we might expect to observe low dissolved oxygen concentrations at the site of the fish kill. Any information or data or analyses that we use to determine whether we actually observe this result, say low dissolved oxygen concentrations at the fish kill site, is a piece of evidence. These individual pieces of evidence gathered across all the results we might expect to observe in our assessment are then combined into the overall body of evidence. This consideration of the entire body of evidence is critical because no one piece of evidence is perfect or tells you everything you need to know. Now going back to the 2 types of evidence I mentioned. In the CADDIS causal assessment process, we think about evidence as coming from the case or from elsewhere. From the case refers to data collected from the specific affected or impaired site and the associated comparison sites in your assessment. These are the data from the sites you're actually interested in. This will be the evidence that is most relevant to your assessment, and it provides the best chance of isolating causal processes and minimizing confounding factors. However, data from the case are often limited, so you also need to make use of what's available from elsewhere. Data from elsewhere refers to data collected from anywhere other than the specific sites you're looking at. For example, this might be data from other field locations, from lab studies, from models, anywhere that's not your site. You can then compare the available data from the case to additional data from elsewhere to derive pieces of evidence. For example, do chemical concentrations at your site exceed established criteria which were derived from other sites? There are 16 total types of evidence in CADDIS, split between those based on data from the case on the left and those based on data from elsewhere on the right here. From the case, the types of evidence you will most commonly be dealing with are the 3 highlighted in blue: spatial temporal co-occurrence, causal pathway, and stressor-response relationships from the field. From elsewhere, the 2 types of evidence you'll most commonly be dealing with are stressor-response relationships from other field studies and stressor-response relationships from lab studies. This doesn't mean you won't ever use the other 11 types of evidence, but the 5 highlighted in blue are the ones that usually have at least some relevant data and are most typically in play in CADDIS causal assessments. Finally, after you've evaluated all the evidence, you're ready to identify the probable cause. In this step, you weigh the body of evidence for each candidate cause included in the assessment. You eliminate any candidate causes you can, and if possible, you diagnose the cause based on specificity of the effect seen, but you're very rarely able to diagnose the cause. You then compare evidence across all candidate causes to determine the most likely cause or causes responsible for the impairment. Evidence is weighed and compared using a qualitative scoring system of pluses and minuses. The full range of potential scores is shown here, but types of evidence differ in the number of pluses and minuses they can get. That is, types of evidence differ in how strongly they can support or weaken the case for a given cause. Each type of evidence has a separate scoring rubric, but the number of potential pluses or minuses a given type of evidence can get assigned is generally determined according to the principles listed here. The first plus or minus, or a 0 if it's very ambiguous, is given based on the logical implication of the evidence you have that passes basic standards of quality and relevance. Data may be limited or of limited relevance or quality, but the first plus or minus indicates that the evidence points in a direction that supports or weakens the case for a candidate cause. The second plus or minus is assigned based on the strength of the association. So, for example, if large differences are observed, the evidence may end up scoring 2 pluses or 2 minuses for certain types of evidence. And then finally, the third plus or minus is based on reliability of the association. So, for example, if sample sizes are high or if there's a strong study design, evidence may end up scoring 3 pluses or 3 minuses for certain types of evidence. And again, it's important to remember that each type of evidence has strengths and weaknesses, and the CADDIS scoring system reflects this. No piece of evidence is perfect, so you want to evaluate as many different pieces of evidence as you can to get the most complete picture possible of what's going on. And I'll show you this scoring system in action for one type of evidence in a few slides. Hopefully it'll make a little bit more sense once you see it, how it actually goes with types of evidence. After the individual types of evidence are scored, the next step is to weigh the evidence for each candidate cause. This is done by evaluating the quantity and quality of evidence, identifying especially compelling evidence, and then evaluating consistency and credibility of evidence. At this stage, you're still looking at all evidence within a single candidate cause. As an example, the table below here shows how consistency of evidence is scored. If all the types of evidence you have for a given candidate cause support the case for it, consistency gets 3 pluses or 3 minuses if all those types of evidence weaken the case for it. It means your evidence is very consistent across all the types you have. If it's less consistent, it can score +1 or -1, and if it's ambiguous or really insufficient, it'll score a 0. Once all the evidence for each candidate cause has been weighed, it's time to compare evidence across causes to form your conclusions. Always compare evidence across candidate causes, even when there's a smoking gun or a cause that really seems to be driving the impairment. This will help you determine if there's more than one likely cause, as well as determine your level of confidence in the results. Do not add up pluses and minuses. Rather, look at all the pluses and minuses across all types of evidence for all candidate causes. Identify the cause or causes best supported by the entire body of evidence and classify all causes, for example, as likely, unlikely, or uncertain, to help identify causes that might benefit from more data gathering and evidence generation. And finally, refine and reiterate as needed. Revise your conceptual model diagrams to reflect your conclusions, as these can be very effective ways to communicate findings. If you have multiple causes remaining, you may need to collect new data and reevaluate and come back and do almost a second tier of a causal assessment. So as needed, continue to refine your findings. Okay. So that was the whirlwind introduction to the stressor identification process, which is explained in much greater detail in Volume 1 of CADDIS. Here's what Volume 1 looks like on the website. It provides information on each of the 5 stressor identification steps organized using 3 self-explanatory tabs: Overview, In-Depth Look, and Results and Next Steps. The different sections under Volume 1 are circled here in red. This navigation bar on the left side of the web page makes it relatively easy to move between these different sections, and this is true both within and across all the CADDIS volumes. Look to the left nav bar for easy navigation through this site. Under steps 3 and 4, there are detailed explanations of all the different types of evidence. So you can see those circled here in red. Again, these are listed under the relevant step on the left nav bar. For example, this is the page for spatial temporal co-occurrence, probably the most common type of evidence you'll deal with in causal assessment. On each of these pages, the concept behind the type of evidence is explained in both words and pictures, and multiple examples are provided. For example, for spatial and temporal co-occurrence, the graphic shows that the case for a candidate cause is supported when impairment occurs where or when exposure to the cause occurs. So depicted here as happy and healthy fish upstream of the discharge and very unhappy fish downstream of the discharge. The case for the cause is weakened when impairment doesn't occur where or when exposure to the cause occurs. In the picture here, you can see unhappy fish upstream and downstream of the discharge and happy fish downstream of the discharge. So this would weaken the case that discharge is causing the problem. Volume 1 also contains handy summary tables of the different types of evidence and how each type of evidence is scored. So, for example, the table at the right shows a portion of the summary table of scores to illustrate potential scores possible for spatial temporal co-occurrence evidence. The highest it can score is 1+, shown in that first row, but the lowest it can score is 3 minuses, shown in the third row. Basically, this means that at most, this type of evidence can only somewhat support the case for a candidate cause, but it can convincingly weaken the case for a candidate cause. So this illustrates how those potential pluses and minuses can be distributed to get across the individual types of evidence. So back to the overall structure of CADDIS. Again, Volume 1 lays out the framework for these steps, and then Volumes 2 through 5 provide information, methods, and tools that can be used at these different steps outlined in Volume 1. And I'll give you a quick tour through each of the volumes now. Volume 2 of CADDIS is Sources, Stressors, and Responses. This volume contains background information, primarily in terms of common stream stressors. There are modules for each of the 13 stressors shown here in black at the right. There's also one source-based module for urbanization at the bottom of this list, which provides an overview of how urbanization affects stream ecosystems. Each of those 13 stressor modules is organized like the module for temperature shown here. Each module has 5 tabs: Overview, When to List, meaning when to list the stressor as a candidate cause, Ways to Measure, Conceptual Diagrams, and References. There are conceptual model diagrams for each stressor on CADDIS, and I encourage you to steal freely from these diagrams for your assessments. This is one example. This is the simple diagram for temperature. There's also a more detailed version with a lot more shapes and lines. And you can see it shows potential linkages between sources at the top of the diagram, the focal stressor in the middle of the diagram, and biotic responses at the bottom. All the CADDIS diagrams follow this general structure. These diagrams provide good starting points for the problem formulation phase of your assessment when you're thinking about what candidate causes and other causal pathway steps you might want to include. And they're often helpful as you're developing the conceptual models for your own assessments in step 2 of the stressor identification process. Volume 3 of CADDIS is examples and applications. These examples include analytical examples that show how data were analyzed for different types of evidence, as well as case studies and other applications of CADDIS in the stressor identification process. To give you a general sense of the information the analytical examples pages provide, here's one of the entries for evaluating spatial co-occurrence using regional reference sites. These pages provide an introduction to the analysis that was conducted, a summary of the data that were used, a description of the analysis conducted and what it found, and then discussion of how the evidence was scored based on that analysis. You can also get to other relevant parts of the larger CADDIS site in the green bordered related information box, and this is true throughout CADDIS. These green boxes are a good place to look for links within CADDIS that may be useful based on the page you're currently viewing. For example, on the analytical examples pages, these links take you to relevant analyses in Volume 4, Data Analysis, and information about the relevant type of evidence in Volume 1, Stressor Identification. Volume 3 also provides examples of how causal assessment has been used by different states. For example, you can see which states have used stressor identification methods in their total maximum daily load programs, the green highlighted states shown in the upper figure, as well as in other applications such as criteria setting or evaluating waste sites or evaluating endangered species. There are also 13 causal assessment case studies at the locations indicated by dots in the lower map. These are good resources for looking at how the entire stressor identification process has been applied in various locations. And in California and Colorado, these case studies are actually for terrestrial environments, so you can see how the principles apply in non-aquatic settings. At least 19 states have adopted all or parts of stressor identification for state-specific applications. Some specific examples include systematically applying causal assessment to watersheds across the state in Minnesota, using causal assessment to inform data collection efforts related to fish health assessments in Pennsylvania, West Virginia, and Virginia, using causal assessments to inform restoration decisions in Connecticut and Maine. And the video link shown at the bottom here describes restoration work in Long Creek, Maine, which was one of the causal assessment case studies included on the previous slide. And finally, working with the Office of Research and Development to make causal assessments faster, cheaper, and routine, as California has done. And I'll come back to this last point about more efficient causal assessments again shortly. Volume 4 of CADDIS is data analysis. This volume contains information and tools on data analysis methods that can be useful for initial data exploration and evaluating the different types of evidence in stressor identification. The main sections of this volume are shown on the right here: selecting an analysis approach, getting started with data analysis, basic principles and issues, exploratory data analysis, basic and advanced analyses, and download software. To give you a general idea of the topics covered in these sections, here are just a few examples. Under Getting Started with Data Analysis, there's information about assembling, matching, and organizing data, dealing with issues of data quality, and links to relevant databases that may be useful in causal assessment. Under Exploratory and Basic Data Analysis, you can find information on a whole slew of statistical topics, from basic information on variable distributions to explanations of more complicated statistical analyses, such as quantile regression and classification and regression tree or CART analysis. There are also more complicated analyses described under advanced analyses. And finally, under download software, you're able to access 3 tools. CADStat, which provides a point-and-click tool for basic R functions useful in causal assessment, things like scatter plots, box plots, correlation analysis, linear and quantile regression, and conditional probability analysis. You can also download a species sensitivity distribution, or SSD, generator. This is an Excel-based tool that calculates and plots the proportion of species affected at different concentrations of a specific stressor, typically as determined in toxicity tests. There's also a module on SSDs in this volume, which explains how they're calculated and used in causal assessment. And finally, you can download an R command line tutorial, which may be helpful for folks that are getting started using R. The last volume of CADDIS, Volume 5, is Causal Databases. This is where the literature-based evidence tools associated with CADDIS reside. There are two components to this volume, or at least there will be two components for the next couple of months. The first is CADLink, which is a database of stressor response information extracted from the scientific literature. The second is the Interactive Conceptual Diagram, or ICD tool, which used to allow you to link this literature-based evidence to conceptual model diagrams that you constructed. The asterisk is here, however, because the ICD has been dying a slow death due to web access changes and the coming obsolescence of Flash. So its current iteration will be decommissioned in the next few months, but we're hopeful we can develop a new improved replacement in the future. So I'll focus on CADLink here. As mentioned, this is a database of evidence extracted from the published literature, particularly looking at specific cause-effect associations. Not surprisingly, it's focused on the associations that are of primary importance in causal assessment. The information extracted in CADLink follows the structure shown at the right here. Each citation may contain one or more data sets. Each data set may provide information about one or more cause-effect associations, and each cause-effect association may be quantified by one or more response measures, or measures of effect size. Fields in CADLink include information on study design, location, and other contextual information that indicate the environment and scales at which the study was conducted, and analytical results. Public users, basically anyone who visits CADLink, can search existing records in the database. Registered users can also enter new or modify existing records. So CADLink can serve as an evidence bank for registered users. And it also allows that evidence to be seen and used by others, making it more accessible for other applications. And you can visit epa.gov/CADLink for more information and instructions on how to register to use the database. So that brings us to the end of the CADDIS Causal Assessment and Stressor Identification Overview and Tour, and brings up the question of what's next for CADDIS. We're currently working on developing methods for more rapid causal assessment in various ways. For example, making the identification of relevant reference sites more efficient, and thinking about how to streamline the stressor identification process. We're also developing the evidence databases through both CADDIS's CADLink database as well as an international collaborative effort called the Ecological Evidence Exchange, or EcoEvidex. The ultimate goal of these database efforts is to crowdsource evidence generation to ultimately improve access to and use of evidence in environmental decision making. We're also working towards linking these evidence databases to interactive visualization tools. We're currently developing EcoDiver, which stands for Ecological Database and Interactive Visualizations of Evidence Records, to allow the interactive display of evidence extracted during a series of nutrient systematic reviews we've conducted. And we hope this prototype will serve as a template for other evidence data sets. We'd welcome any and all suggestions for what improvements or additions you would like to see. So if you have ideas, please contact us. Let us know what's been helpful, what hasn't been helpful, what might be helpful in CADDIS. We're also always looking for states to partner with as we work on these different components of assessment, so please get in touch if you have ideas for collaborations that you think would be useful. Finally, I want to conclude by going back to a point I made early on, which is that much of what I've talked about today, although originally developed for causal assessment of stream biological impairment, is really more broadly applicable. I've touched on this a bit already in that the CADDIS stressor identification process has been used to look at impairments in terrestrial systems and hospital sink drains. But these same ideas and tools also can be useful in other types of assessment. For example, the schematic here shows the process I've talked about today, causal assessment designed to figure out which stressor or candidate cause is likely causing some observed biological impairment. But many of these same ideas, types of evidence, and tools can be useful in trying to assess causes of things other than biological impairment. For example, increases in a particular stressor like phosphorus concentration. Here, the candidate causes of the effect of interest are different sources, runoff, atmospheric deposition, remobilization from sediments. And you can use the same types of evidence to evaluate these hypothesized pathways. What may be the endpoint of interest in one assessment could be one of many candidate causes in another assessment, but the same principles and methods often can be used in both cases. So CADDIS is a website that was originally developed for causal assessment, but that does not mean that it's the only kind of assessment it's useful for. Thank you for your interest and attention today, and if you'd like more detailed information on any of these topics, please get in touch. We often give multi-day CADDIS training, so this presentation really just provides a quick intro.